Okay, all I'm planning to do today is finish chapter 6. I know that probably puts me behind schedule, but I can make up in at a future date. Tuesday is a holiday, it's Mardi Gras, and so the next Zoom meeting won't be until next Thursday. And before I leave today, I'll post the link to the recording that I generate today. And so this is simply the rest of chapter 6. I'm about, I guess, two-thirds of the way through. And this is going to be genomic imprinting, which is kind of weird, and maternal effect inheritance, which is also kind of weird. Anyway, this should be where I started, where I left off on Tuesday. And so in genomic imprinting, there's modifications to a gene that alters its expression, but this imprinting is not going to be permanent. An expression of a gene can depend upon whether or not that gene is inherited from the male or the female parent. And phenotypes controlled by imprinted genes have a very non-Mendelian pattern of inheritance. And in genomic imprinting, the offspring will express either the maternally inherited or the paternally inherited allele, but not both of them. And so this is monoallelic expression. But me, it helps if I do that. And so this is epigenetic inheritance. And epigenetics means patterns of inheritance that exist above the level of the genotype. And so this is some kind of a modification to a gene. It changes gene expression, but again, is not permanent. Now, it might have a permanent effect on the life of an individual, but it is not a mutation. It is not a change in the DNA sequence all by itself. It's just a difference in expressivity. These changes can be inherited, but the inheritance patterns are not predictable through Mendelian genetics. Now, these chromosome modifications can occur during gametogenesis, or they can occur early in development. And an easy example in mammals is X chromosome inactivation. Another example is what they call imprinting. And so in imprinting, there is going to be some kind of modification to a gene that's going to change the expression. But again, it is not permanent. Expression of that gene depends upon which parent it is inherited from. And so again, you'll express one or the other, but not both. There is an example in a lot of mammals. There is a gene that encodes a protein. It's called IGF-2, insulin-like growth factor 2. A functional version of this gene is necessary for normal size in the mammal that has it. And for whatever reason, the imprinting has resulted at this example in mice. You express the allele you got from your father, but not the allele you got from your mother. And so the paternal allele is transcribed into RNA, which is then translated into a protein, and nothing happens at the maternal allele. And IGF-2 minus is a knockout mutant that causes a defective protein to be made. And that will make in this example, the mice to be dwarf. And this is only true if you inherit a IGF-2 minus allele from your father. So there is the example. You have a mother whose genotype is IGF2 minus, IGF2 minus. So she is a homozygous recessive. She will be dwarf. The father's genotype is IGF2, IGF2. He is homozygous dominant. He will be normal sized. All the offspring will be heterozygous, but they will have inherited the IGF2 normal allele from their father and the IGF2 minus allele from their mother. All of these heterozygotes will be normal sized. And if you do a reciprocal cross, the mother genotype is IGF2, IGF2, and the father is IGF2 minus, IGF2 minus. Again, you will make a bunch of heterozygous individuals, but this time all of these heterozygotes will be dwarf because they inherit that IGF2 minus allele from their father. That one is expressed, and the unmutant one is not. Now, in that example just given, the F1 offspring have the exact same genotype. They are all heterozygous. At the phenotypic level, they are different. So even though they have the exact same genotype, they're going to look different because they got one gene from the father and one gene from the mother. Because of this, unless you're very careful, you cannot easily use a Punnett square to predict an offspring's phenotype when these genes are imprinted. And you have to know some things to make an educated guess. Which allele is expressed? Is it from the male parent or the female parent? And then you have to know which allele was inherited from the mother and which one was inherited from the father. Now, you can construct a Punnett square that will allow you to make a guess, but then you actually have to do the physical cross to see your results and then see which combination of inheritance patterns match the phenotypes that you observe. And so imprinting can be divided into a few stages. During the formation of gametes, the imprint gets established. During embryogenesis and throughout the adult life, that imprint has to be maintained in the somatic cells. And then in the germline cells, the imprint will get erased and reestablished. And so the gametes will be made with the imprint in place. And so establishment, sperm cell, egg cell. And in this case, you get the IGF2 normal copy from the male and the mutant from the female. And so all of the mice born in this generation, heterozygous, but normal size. In the germline cells, this imprintation gets erased and then reestablished. And so in the eggs that are produced, that particular gene has been silenced. In the sperm cells that are produced, that gene is active. But one half of those sperm will carry the mutant version. The other half will carry the normal version, and that will affect the inheritance pattern. Genomic imprinting is permanent in the somatic cells of any given generation. However, this imprinting pattern can be altered from one generation to the next. Imprinting occurs in most species we have examined, and it includes mammals, insects, and plants. The imprinting might involve a single gene, a part of the chromosome, an entire chromosome, or all of the chromosomes that you get from one parent or the other. And so the extent could be anything. And imprinting typically involves some way to mark specific regions of the chromosome. And a very common way to mark specific regions of a chromosome is to methylate certain bases. And you just means you add methyl groups to these bases. And we call these as imprinting control regions. Those control regions tend to be near the imprinted genes. The methylation usually causes the genes in the area where they're methylated. It usually causes the DNA to be overwound in those areas. The methylation will occur in one germline cell or the other. And so it will be in the oocyte or the sperm, but not in both. And these ICRs contain what they call a transcription factor binding site. And if DNA is overwhelmed in that area, transcription factors cannot bind, and so you don't make mRNA from the DNA. And so here's your somatic cell. And this methylation will occur in every somatic cell. During gamete formation, this methylation gets removed, and then you make normal gametes. In the opposite situation over here, the start of gamete production, the methylation gets removed, cells divide, but then the methylation is reestablished. And so the sperm cells all will carry methylated versions of that allele. The importance of it. A few genes have been shown to be imprinted in humans. 100 genes is not all that many. And so there are a few genetic diseases that we have traced back to imprinted genes. And here's a list of them. And there's the gene name, the expressed allele from what parent is expressed. And for example, this is a Wilms tumor suppressor gene, which suppresses cell growth. And then you have, gene that encodes insulin, like growth factor, etc. And just for whatever reason, you express the allele you get from one parent or the other. And the maternal effect, it was the easiest example, best known example is the type of coiling in a snail shell. And in maternal effect, only the genotype of the mother affects the phenotype of the offspring. The genotype of the father has no effect. The genotype of the offspring has no effect. And this maternal effect is controlled by nuclear genes. And we can trace that back to when the egg cells are developing, the egg cells will be surrounded by support cells, and these support cells will provide gene products to the egg cells that are developing. And the egg cells that develop will express the RNA they get from the surrounding quote unquote nurse cells. And so there was coiling in this snail, discovered back about 100 years ago. And these snail shells can be a right handed twist, which is called dextral, or a left handed twist, it's called sinistral. The thing is, if you're a snail, the way your shell coils determines the alignment of your internal organs because they have to follow the way that shell is twisted. And so crosses of those F1s demonstrated pattern of inheritance that was not explained by Mendelian genetics. And here are some sample crosses. You have a male parent with a dextral twist, female parent with a sinistral. Homozygous for both of them. Bunch of offspring, all heterozygous. In this case, all dextral. And you do the opposite. Female parent that is sinistral, male that is dextral, offspring, all heterozygous, all sinistral. Across individuals of this generation, you get a normal 1 to 2 to 1 genotypic distribution. But in this case, since the female parent was going to be, since the female parent is heterozygous, when her eggs are formed, the surrounding cells will contribute at least some of the RNA product from this dextral allele. And so the eggs that are made will all have this dextral twist. If you took this one generation further, you would get the F3 generation, and you would have a 3-to-1 ratio, dextral to sinistral, and it would be more or less equally either a right-handed twist or a left-handed twist in snails of either gender. And so, easily explained. When this was discovered, it was proposed that the shell coiling was controlled by what they call a maternal effect gene. The phenotypes in the next generation are controlled by the genotype of the mother and genotype of the father, or the members of that generation, that F1 generation, don't really make a difference. If the mother is capital D, all the next generation will be dextral. If the mother is a heterozygote, all the next generation will have the dextral twist. If the mother is homozygous for the sinistral, all the next generation will have that sinistral twist no matter what their genotype. And the thing is, and it's easier to show here. Here's a developing egg, and here are nurse cells surrounding it. And if these nurse cells contain at least one of these capital D alleles, that RNA will get pumped into the egg. And that is enough to make that offspring have a dextral twist. And once that cell in the offspring starts to divide and you form a body plan, it is going to force the rest of the organs in that snail to follow that body plan. And so here it is. Any animal gamete is going to be surrounded by nurse cells. And in the case of the oocytes, these developing oocytes are surrounded by cells from the mother that are going to provide them nutrients. The nurse cells are diploid. The oocyte will become haploid. If that female is heterozygous for the snail coiling gene, this capital D allele product will be put into the developing oocyte. And that will cause that right-handed turn. If the female is nothing but recessive little d, all that we put in the developing oocyte is the gene product from that little d. And that will cause a left-handed turn. And so basically, if the mother is capital D, the egg will get nothing but capital D's. If the mother is heterozygous, both gene products can be put in. But since the capital D allele is dominant, it will express itself over lowercase allele. And if she is homozygous for little d, the developing egg will accumulate only the protein product from little d. And so all the offspring will be sinistral. And so there is basically the way that snail embryogenesis happens. It has a fairly peculiar kind of division in the embryonic cells. Now, maternal affected genes work because they encode RNA, therefore proteins, that they pump into the egg cell that play a very important role in the very first few steps of embryogenesis. And they determine what, for example, what kind of cell division the early embryo undergoes. They can determine the body axis that the early embryo must therefore follow. And they determine the cleavage pattern in the early few rounds of cell division. And what happens? These proteins and whatnot get pumped into the oocytes. And that allows the development to happen very fast. So the first few rounds of cell division are very fast. In the fruit fly, geneticists have identified a few dozen maternal effect genes. And they tend to have pretty profound effects on how that organism goes through the first few rounds of cell division. And after the first few rounds of cell division have been, done, the later stages of embryogenesis must follow what the earlier stages of embryogenesis established. And like I said, I want to end there. It is probably too slow. I'm going out of town for reasons on Saturday. I'll be back on Monday. Monday and Tuesday are holidays. And so I will be back on campus next Wednesday. Um, I'm trying to stop using Canvas, sorry, Connect for homework because I know that the bookstore marks the cost of the textbooks up. And so to get access to Connect, you have to buy the book. And that might run a few $100s. And so I have to think about assignments that I can post straight to Canvas and not have you work through Connect to access. And that will mean that everybody, since I post the assignments to Canvas, that means everybody will have access to the homework. And I'm going to have to think about assignments to give because, in all honesty, for the last couple of 3 years, I've used Connect pretty much for 100% of the homework points. And it's been since before the pandemic that I use something other than 100% Connect. And so I'm going to have to do some searching on my computer to see what kind of things I can define, what I can find. Okay, I'm going to end that now. I should have a student from another class coming to take an exam. Um, I hope she remembers. And so I got to wait for her.